Imagine your gut bacteria whispering to your DNA, turning genes on or off without changing the code itself. This is epigenetics in action. Epigenetic studies how external signals alter gene activity without changing DNA. DNA methylation adds a methyl group to cytosine and CpG islands. This chemical tag can silence nearby genes by blocking transcription factors. Histone proteins package DNA into nucleosomes, forming chromatin fibers. Acetyl groups attach to lysine residues on histones, loosening the fiber. Looser chromatin allows RNA polymerase easier access to gene promoters. Conversely, methylation of histone tails can tighten chromatin and repress transcription. Non-coding RNAs guide protein complexes to specific genomic loci. MicroRNAs bind messenger RNAs, preventing their translation into proteins. Together, these mechanisms create a dynamic epigenome that responds to the environment. Epigenetic marks are inherited through cell division but can be reversed. Understanding these processes helps explain how microbes influence host gene regulation. Pause 0.8. DNA methylation begins when a cytosine in a CpG dinucleotide receives a methyl group. DNMT enzymes attach the methyl group using Sa-urose adenosylmethionine as donor. The primary maintenance enzyme, DNMT1, recognizes hemethylated DNA after replication. During synthesis, the new strand is unmethylated while the parent strand stays methylated. DNMT1 binds to the unmethylated cytosine and transfers a methyl group from its partner. This copying restores the symmetric CpG pattern on both strands. Because the pattern is Ra-urose established each cell cycle, repression becomes stable. Genes flanked by dense CpG islands often remain silenced through this mechanism. Heat maps reveal that different tissues display distinct methylation landscapes. Such tissue-urose-specific patterns Urose tune gene expression during development and differentiation. Pause 0.8. Histone tails extend out of the nucleosome core, ready to receive modifications. Acetylation neutralizes positive charges on lysine residues, loosening DNA binding. When a histone deacetylase removes an acetyl group, the tail regains charge. This increased charge tightens the DNA wrap and reduces accessibility for factors. Methyl groups added to arginine or lysine can either activate or silence genes. The specific pattern of methylation determines whether a transcription factor is recruited. Phosphorylation introduces negative charges, causing subtle structural shifts in chromatin. These shifts can expose binding sites for remodeling complexes that reposition nucleosomes. Ubiquitination tags histone tails for recognition by the proteasome or other readers. Readers interpret ubiquitin marks to either promote transcriptional activation or repression. Together, these modifications orchestrate a dynamic chromatin landscape that governs gene expression. Pause 0.8. Bacteria in the gut produce small molecules that can reach host cells. One key product is butyrate, a short urose chain fatty acid generated by fermentation. Butyrate travels across the intestinal epithelium and enters colonocytes. Inside these cells, it inhibits histone deacetylases, or HDAC. HDAC inhibition prevents removal of acetyl groups from histones. The resulting increase in histone acetylation loosens chromatin structure. Looser chromatin allows transcription factors easier access to DNA. Consequently, genes involved in barrier function and immune regulation are urose regulated. Secondary bile acids produced by bacteria also target epigenetic enzymes. They can modulate methyltransferases, altering DNA methylation patterns. These epigenetic changes illustrate how microbial metabolites shape host gene expression. Pause 0.8. Bacteria release small signaling molecules that travel through the local environment. One such molecule, AI2, is produced by many species in a shared language. Anichyl homoserine lactone molecules bind to dedicated receptors on the surface of host cells. Binding initiates a signaling cascade that transmits information into the nucleus. Inside the nucleus, transcription factors recruited by the cascade recruit DNA methyltransferases. These enzymes add methyl groups to cytosine bases and promoter regions of immune genes. Simultaneously, histone acetyltransferases and deacetylases modify chromatin structure. The combined epigenetic changes either enhance or suppress transcription of target genes. As a result, the host cell adjusts its immune response to the bacterial presence. This dynamic interplay illustrates how microbial signals can shape host gene regulation. Understanding these mechanisms provides insight into the coevolution of microbes and their hosts. Pause 0.8. The microbiome is a dynamic community that can influence host epigenetic marks. Microbial metabolites, such as short-chain fatty acids, act as cofactors for DNA methyltransferases. When butyrate levels rise, DNMT activity increases and cytosine residues become methylated. Conversely, microbial production of histone deacetylase inhibitors can loosen chromatin structure. These chemical signals are transmitted through the bloodstream to distant tissues, including the gut epithelium and liver. Over time, repeated exposure creates a stable pattern of DNA methylation that can persist across cell divisions. Dietary shifts alter the microbial composition, which in turn reshapes the epigenomic landscape. A composite heat map can overlay microbiome diversity indices with host methylation profiles to reveal correlations. A timeline graphic illustrates how these changes evolve during successive dietary interventions, highlighting causality. By integrating microbial and epigenetic data, researchers can predict tissue-specific responses to environmental cues. Pause 0.8. Epigenetics allows cells to modulate gene activity through reversible chemical tags on DNA and histones. Gut microbes supply metabolites like butyrate that inhibit HDAC, increasing acetylation, while quorum sensing molecules can trigger methylation changes in host genes. Together, these interactions create a dynamic epigenomic landscape shaped by microbial community composition.